Hello, everyone. My name is Aspasia Vazquez. I'm an undergraduate research student. I'm an undergraduate student at the University of Colorado Boulder. I'm majoring in ecology and evolutionary biology, and I'm minoring in geology. And this summer, I did a uh, NASA YSP internship with Dr. Elta Howells, and my project is examining methanogen protein adaptation to extremely low CO2 availability. So some of the environments that we study are hyperalcophilic environments. Hyperalcophilic environments are very high in hydrogen, very high pH, and very low in CO2. And these environments are a very good analog for like other ocean worlds and that might be in our solar system, like Enceladus. And some of these environments also house microbial life. Like this in the picture, we can see this is a serpentinized fluid hyperalcophilic environment which is the Somali opalite in Oman. The opalite in Oman has serpentinized fluid that is rich in hydrogen. It's hyperalkaline and it's poor in DIC, which is dissolved inorganic carbon. The figure to your left is a figure kind of displaying how the mixing in this environment occurs. There are various ranges of pH in these environments, and so we can kind of see here that there is a pH 8 kind of surrounding surface environment, and those are our type 1 serpentinized fluid serpentinized fluids. And then we have like a pH 11, which is our type 2 serpentinized fluid, which is much higher in pH. Our type 1 fluids are closer to the surface and have a lot more interactions with CO2 and have a lot more atmospheric interactions as well, while our type 2 fluids have way less and are very low in CO2. And within this, in the pool, there is also mixing between these two environments under the subsurface. Um, uh, because of this, we get to see a very beautiful gradient within this environment. So we get to see a cool CO2 gradient within one geographical location, which is important for study. In the methanogens that live inside of this environment go through a metabolism process called autotrophic methanogenesis. I would like to highlight the equation at the bottom, which is just is the equation for autotrophic methanogenesis. And as we can see, hydrogen is important in this system. Since the system is very high in hydrogen, they have way plenty enough hydrogen, but they don't have as much CO2, which is the other element of this. And so we were hypothesizing that CO2 availability might be important. Here we can see a figure kind of displaying the differences between a serpentinized environment and then you're like your typical environments where you might find methanogens. The top like red figure on the top of that figure in the middle of the screen is your marine and freshwater sediment environments, while the green square at the bottom is our serpentinized fluid environments. It is on a scale of hydrogen and CO2. We also see that there is like a huge difference between these two. There's actually an 8 orders of magnitude scale here, which is millions and millions and millions of molar difference, which is quite a lot. What we presently know about methanogens can't explain their occurrence in serpentinized fluid, but we know that we find them here. And in my summer internship this summer, I focused on an enzyme that carries out the first process of autotrophic methanogenesis that I explained earlier. The enzyme that I particularly studied is called formylmethanoferrin dehydrogenase. And the first figure I'd like to highlight on the screen is the one to the left. This is a diagram that outlines methanogenesis. And as you can see, one of the first steps of methanogenesis is to take in CO2 from the environment and then process it into formylmethanoferrin and then continue on through the process. But I would like to focus on this enzyme that does this. And as you can see to the left, this is a 3D crystal structure of FMDH, or from formylmethanoferrin dehydrogenase. Something very specific that I pointed out, that I would like to point out and also focus on in my study, is that there are multiple subunits present in this enzyme, and that the blue region is the FWB subunit, which houses a CO2 tunnel, which allows it to take in CO2. And then the CO2 will travel down to the first active site, become formate, and then go to another tunnel called the formate tunnel in the subunit A, FWA, which is the green area of the crystal structure. And then it will go to another active site at the end of the tunnel and turn into formylmethanoferrin. I'm next going to talk about FMDH phylogenies that I created with these subunits, and then also a protein metastability analysis. This is the first FMDH phylogeny that I created. It's a maximum likelihood phylogeny of the sequences that for FWB. So these are our CO2 tunnel sequences. The thing I would like to point out with the phylogenies very specifically is that we had many different kinds of microbes that we had in our studies. Some were sampled from Oman, and some were just sampled from taken from other studies or from other environments. But what I'd like to point out specifically is that all of our hyperalcophilic environment methanogens formed one singular clade, which is that clade on the top in the green circle. And then at the bottom, we have another clade, which is all of our neutrophils, which are like the opposite of a hyperalcophile. And I can point out very specifically the three spetnized fluids that some of our methanogens came from. Our type 1 fall into our neutrophilic environment because of their pH and CO2 availability, but we also have a mixed and type 2 environment here. And as you can kind of see, they all have formed distinct clades based on CO2 availability. We can also highlight this figure in to the bottom right, which talks about on axes of pH and molality, you can kind of see the differences in these environments. There's a lot of different points on this figure, and each one is like a specific sampled like analysis, but what we can see and what I've labeled is that there's a type 1, a mix, and a type 2. And as you can kind of follow, all of the diamond points on this figure represent CO2. And so you can kind of see how the CO2 changes within these types of serpentinized fluids. I also did the same analysis for my tunnel, and I saw a very similar thing happening as well. There was a same kind of distinct clades forming. We also did an assessment of the amino acid composition within these FWB sequences. And we kind of saw that there was both hyperalkophiles and the neutrophils had even abundance of polar uncharged and nonpolar hydrophobic amino acids. Neutrophils, though, had a more abundance of acidic amino acids and that have more N2 in their functional groups. What we know is that there are differences in the amino acid composition of FWB between neutrophil and hyperphilic based on what we saw previously, but we wanted to investigate more why that is because we couldn't necessarily tell just on face value. So what we hypothesized is that CO2 in the carboxyl group containing amino acids would be driving factors for these differences. And we've used a program called SNAS to do an assessment estimating thermodynamic properties of chemicals and reactions, including the proteins. I would like to highlight specifically the two amino acids that we focused on for this within the FWB sequences, which is aspartic acid and glutamic acid. As you can see in these two figures at the bottom of the screen, aspartic acid and glutamic acid both contains a carboxyl group, which is made up of CO2 and other things, and that there are distinct differences between our neutrophil and our hyperalcophile. For this study, I would also like to note that we chose one hyperalcophile and one neutrophil methanogen for this study. To estimate these, we needed to look at them as if they were a compound because this SNAS is a type of program that is generally used not for this purpose. And so what we decided to do is a little bit more experimental and not necessarily done as much. And so what we did is we kind of broke them down into what they are made up of, which is carbon, hydrogen, nitrogen, oxygen, and sulfur, which is reflective in their environment. And so as you can kind of see here between our neutrophil and our hyperalcophile had less of both carbon and oxygen than our neutrophil, which is reflective for its environment as well. Here we have an example of a DIC species graph. This compares pH and concentration for DIC, which, as I briefly mentioned a little bit earlier, is a dissolved inorganic carbon. Minerals like these, which is carbon dioxide, bicarbonate, and carbonate, that, as you can see on the graph, and we can use these analysis like this to understand the geochemical process and how it interacts with the environment. DIC species equations are typically a sum of aqueous species of inorganic carbon in an environment that are at a dynamic equilibrium, meaning they're always interacting between one another, and their interactions are influenced by environmental factors such as concentration of the compounds, pH, temperature, and also just other factors as well. And what we can kind of see from this is an example of one of those equations. And so we kind of took this and tried to apply it to our own city. Keeping with that, we kind of took these two sample species that we looked at, one neutrophile and one a hyperalkalophile, and we looked at them if they were compounds, we made an equilibrium reaction for them as if you were doing a DIC species analysis. And this is a very unique approach and one that we kind of experimented with, and it's not really usual, but we use schnoz to help us with this. And I'd like to highlight the carbon differences between these two mags. The carbon value does not directly correlate to the amino acid sequences, but they do demonstrate a point about carbon availability in the environment. Our neutrophile, for instance, has more carbon than our hyperalkalophile, which is reflected in their environment. And we're seeing it reflected as well within their sequences, which is quite interesting. And these are also very preliminary techniques and results for this as well. And here are some graphs from that same protein mendability sequence analyses. And what we kind of did we did two types of studies, and this was the first one. We compared a neutrophile and a hyperalkalophile for this, and this just models CO2 activity. And activity, think of it as proportional to concentration. So of CO2 in the environment. And so what we kind of see here is that our neutrophile has more CO2 activity than our hyperalkalophile, which we would expect based on their CO2 availability. And we did two replicates of this and saw two very similar results. We did another study between our one strain of each type of serpentinized fluids, one type 2, one mixed, and one type 1. And we saw something very similar as previously that we would expect, where our type 1 had more CO2 activity than our type 2, and our mix was somewhere in between. As well, we did two replicate studies for this and saw something very similar with both, which can be also reflected in their environment as well. I would also like to highlight that graph I had up earlier, once again, just to kind of further demonstrate the CO2 availability. And you can kind of see it and here. We have our type 1, our mix, and our type 2. Overall, neutrophilic FMDH have higher relative activities under conditions with relatively higher CO2 activity. The opposite is true for hyperalkophiles, though, and a low CO2 confers with a low FMDH activity. Some of our next steps that we thought of with this analysis was to add more methanobacterium to our analysis to kind of expand our pH range and be able to make it applicable to a more broad range. And we also would like to sequence and add an NSHQ field culture to our analysis and investigate further selective pressures like pH or CO2 in this environment. And these are preliminary results, and we would like to eventually investigate this further because we got some very tricky results. I would like to acknowledge my mentor, Dr. Alta Howells, for allowing me to do this project, and special thanks to NASA Ames Research Center, the GOM Lab at CU Boulder, and very special thanks to the Blue Marble Space Institute of Science for allowing me to do this talk today, and for setting up this program and allowing me to participate. I will now open for questions. Thank you so much.
Awesome. Great job, Aspasia. Having been somewhat involved in the hydrogen generation research and some of the research in Oman, I was in Alexis Templeton's lab as a graduate student years ago, having seen many versions of the figures of the type 1 and type 2 fluids, and it's all really incredible. This brought me back so much to the research I've done myself over the years. I see Sanjoy has a question about your presentation. I'll let Sanjoy start off questioning here.
Yeah.
Hey guys, very nice talk.
Oh, thank you.
So FMDH can only handle carbon as CO2 on its way to methane. And yet in serpentinizing environment, like you mentioned, those sites are challenged with CO2 because of the de-icing speciation challenges. I find it surprising that biology has not figured out a way, especially for alkalophiles, to use carbon in other forms than CO2. Why do you think it's still um, hampering itself by forcing the use of CO2 as a carbon source?
Um, I'm not entirely sure, but I could probably think of some kind of like maybe possibility of just like its interaction with the atmosphere or something like that. I would have to do some probably more research into that, but that is very interesting, and I wonder that as well.
Yeah, it's curious. Yep. If you find anything intriguing, do let me know. I'm sure Alta has a lot of ideas as well.
Oh yeah, of course. Yeah, that'd be great to look into.